Hello guys, today I am going to discuss a very important application of flow cytometry, that is how do we analyze cell cycle by using a flow cytometer. Today I am going to explain mainly the propidium iodide method by which we can identify that how many cells in a given population are present in G1 phase, how many cells are present in S phase, and how many cells are present in G2 phase, and how we can utilize different situations to understand the effects of different drugs on cell cycle of a particular cellular population. So before jumping directly to cell cycle analysis, I will give you a brief idea about the basic principle of flow cytometry. So the word cytometry uh, is used when we are measuring physical and chemical properties of single cell. So what are the physical properties, what are the chemical properties, what are the proteins present in that particular single cell? When we are calling the whole thing as cytometry. And when I am adding the word flow in front of cytometry, then the flow cytometry is defined by the statement that we are measuring the physical and chemical properties of single cell, but we are using a fluid system or fluid stream in an instrument called as flow cytometer. So only one thing which I am adding here is a stream of fluid. So let's understand what I mean by the fluid system, or what a flow cytometer is actually doing when we are putting our cells in the instrument. So if I look at aspect the functional aspect of the instrument is very simple. It is taking single cell suspension, and by using the pressure of fluid, so it is the flow of fluid, and by putting or by applying the pressure of this fluid, the instrument is converting these bunch of cells in a system where one cell is passing at a time. So it's very simple. There are so many cells. You apply the liquid pressure from both the sides. So you can create an array, or you can create a stream of buffer or stream of a liquid where one cell at a time is passing through. And that one cell is interacting with laser, the light source. And these interactions are measured by different kind of detectors. The principle is very simple. And this application of pressure and generating this stream is called as hydrodynamic focusing. Very simple to understand. You take some cells, you apply hydrodynamic focusing, you apply the flow of liquid, a pressure, create a single array of cells. Then one cell at a time is interacting with laser and giving us some information which is detected by different type of detectors, right? Nowadays there are flow cytometers which are coming with acoustic sound waves, where along with hydrodynamic focusing, a sound wave is also being utilized to create this array of cell, or to create this situation. So the principle is not in the working of instrument, but principle is lying mainly where we have to understand what kind of interactions are happening between the laser and the cell. So let's understand that. Let's understand the interaction of light with the cell. So here it is our light source, the laser. Then we are having incident light beam, and here is our single cell. So if the cell is not coming, then the laser is easily passing through the liquid, because there will be only buffer, there will be no cell, and it is detected by detector as no event. And if a cell is there, then the cell will interact with light, the light will be scattered by this cell, and the machine is counting it as one event. second cell means second event, third cell means third event. It is going like that. So absence of cell, no events, and presence of cell, yes, there are events. Now, the light can interact with the cell in three manners. The first manner is the light can scatter from the edges of the cell, and the angle of the scattering is between 0.5 to 5 degrees, and we call it as what? Forward scatter. What do you mean by forward scatter? The light is coming, interacting with the cell, scattered between the angles of 0.5 to 5, and this scattering is occurring because of the edges of cells, right? We are calling it as what? Forward scatter. And the forward scatter is giving us information about the size of cell. So if we are having bigger cell, then of course the scattering will be higher. The value of forward scatter or FSC will be higher. So if the instrument is telling us that for a particular event FSC value is this, that means that is the indicator of cell size. Higher the cell size, bigger the cell size, the value of FSC is high. Now, some of the light can enter inside the cell and it will be scattered by the organelles, by the biomolecules, or you can say by the granules of the cells. This kind of scattering is between 15 degree to 150 degrees, and we call it as what? Side scatter or SSC. And we already know now that the value of SSC is giving us information about internal granularity of the cell. So more granules, higher the value of what? Side scatter. Bigger the size, higher the value of what? FSC or forward scatter. So the FSC and SSC, two parameters, forward scatter and side scatter, are giving us the information about size of the cell as well as granularity of the cell. The third information which we can get from a flow cytometer, if my cell is containing a fluorescent protein like green fluorescent protein, red fluorescent protein, or stained by a fluorescent dye like propidium iodide, cyper green, cytox, or the proteins of this cell are labeled with fluorescently tagged antibodies. So it can have an inherent fluorescence, it can have presence of a fluorescent protein, it can be stained by a fluorescent dye, or it can be having fluorescently labeled antibodies which are binding against specific proteins of this cell. So all these type of fluorescent molecules can absorb the light at a particular wavelength, and then emission of light will happen, and then ultimately we will get the detection or we will get the information. So three types of information. First, forward scatter size, side scattered granularity, and finally presence or absence of a fluorescent molecule, right? Now we have understood that we can understand the size, granularity, and fluorescence and how the instrument is working. We can see or we can understand how the flow cytometer is giving us the information about cell cycle or giving information about how many cells are in G1, how many cells are in S, or how many cells are in G2. So this is our cell cycle divided into phases of G1, S, G2, and M. So what kind of fundamental I require to understand the flow cytometry based analysis of cell cycle? The answer is very simple. I have to look at the cell cycle from the point of view of DNA content. It means in which phase of cell cycle how much amount of DNA is present. So guys, now we understand how we can analyze the cell cycle or its different phases by using flow cytometry. So we are having phases of cell cycle, that is G1, S, G2, and M. But to analyze it with the help of flow cytometer, we have to look at the cell cycle from the point of view of DNA. What does I mean? What do I mean? The answer is very simple. In G1, let's say the cells are from human. So we know that in G1 phase, cells are resting. They are preparing for cell cycle. So they are containing 46 DNA molecules, right? 46 chromosomes or 46 DNA molecules. Now in S phase, they do replication, and in G2, before G2 the replication is completed. So of course in G2M, we are having 92 DNA molecules. So a particular amount of DNA, that is X, in G1, and then in G2 it will be 2X, and in the S phase it will be between X and 2X. Why between? Because if the cell has just started the replication, then the amount will be different. If the cells are in between the replication, then the amount will be different. And if the cells are at the end of replication, the amount will be different. So how much amount of DNA will be there in S phase between X and 2X, or between 46 DNA molecules and 92 DNA molecules, is depending upon at what stage of replication the cells are, or how much percentage of replication is completed, right? So let's do the experiment by using the flow cytometer, and we will be understanding it better. But till now, G1, X amount of DNA, G2, 2X amount of DNA, and S, the DNA amount is in between X and 2X. So let's start the experiment. So if I take out the cells from a culture flask, a population of cells, let's say I am taking 5 million cells, then I am preparing a single cell suspension from this flask, and in this single cell suspension I am putting a DNA binding dye, and after incubating the cells with DNA binding dye, I am putting the tube into our flow cytometer, and then flow cytometer will give us some kind of graph, some kind of analysis. Now what is that kind? So again we are having the cell cycle, and this time we are having multiple cells. Now some of the cells will be in which phase? G1. So let's say out of 5 million cells, 30 to 40% cells or 50% cells are in what? G1. Then some of the cells will be in the S phase during replication or doing the replication. Let's say 10 to 20% cells are in S phase, and some of the cells are in G2M. So it means I can say that these cells are having X amount of DNA. These cells are having the amount of DNA between X and 2X, and these cells are having 2X amount of DNA, X, 2X, and in between. So what kind of graph I will be getting? So what I am using here, DNA binding dye. So I can safely say that these cells will be having the least amount of DNA binding dye, or this DNA binding dye, one of the example is propidium iodide, which give us fluorescence after the binding. So I can say that the cells which are present in the G1 will be giving us least amount of fluorescent signal. Then these cells will be giving us higher amount of fluorescent signal than G1, but lesser than G2 and M. And the cells which are present in G2 will be giving us highest amount of fluorescent signal, because the DNA is X, 2X, and in between X and 2X. So the binding of DNA binding dye will depend upon how much DNA is present inside the cell. So the graph which we get is like this. On y-axis we are having number of cells, and x-axis we are having DNA content. So when I am moving right, then amount of DNA is increasing. So of course this DNA content will be measured by fluorescence given by DNA binding dye. So these cells which are giving us least fluorescence, they are present in what? G1 phase, X amount of DNA. These cells which are giving highest fluorescence, one more time, highest fluorescence, then they are going to give us the amount of cells or percentage of cells which are present in G2M. X amount of DNA, 2x amount of DNA, and these cells are present in S phase. Why we are not getting a single line, we are getting distribution, because some cells are at the starting of S phase, means they have just started the replication, means their amount is slightly higher than X. Then here the amount of DNA is increasing, and ultimately slightly lower than 2x and finally 2x. So multiple cells can be present at different stages of completion regarding replication. So if they have just started the replication, amount of DNA will be less, fluorescent signal will be less. If they have ended the replication, then the cells will be present here. And if they are in between the replication, the cells will be. So this type of graph we are going to get in the flow cytometer. We can select the area and we can calculate the percentage of cells. So you can find out what is the percentage of cells present in G1 phase, what is the percentage of cells present in S phase, and what is the percentage of cells which is present in G2M phase. Now what kind of result I actually get in a flow cytometer? This is the graph. Very simple to understand. We are having the peak of G1 phase, X amount of DNA. Then we are having the peak of G2 phase, 2x amount of DNA, that is diploid 2n, then double diploid, that is 4n. And in between we are having the cells which are in S phase of cell cycle or doing replication. Least amount of fluorescence, FL2 is the channel of flow cytometer, which is giving us the fluorescence of propidium iodide. Least binding of DNA binding dye, least amount of fluorescence, that is X. Highest binding of propidium iodide, highest amount of DNA content, highest amount of fluorescence signal, 2x. In between, signal is between X and 2x, or amount of DNA is X and 2x. Now you can see there are some cells which are less than G1 phase. These are the cells which are undergoing apoptosis. They are fragmented. So they are containing DNA amount lesser than what? G1. So we are getting so many cells here. We are getting some cells here also. What do you mean by that? They are having amount of DNA more than 2x or more than G2. So these cells can be diploid, means these cells can be doublets, means two cells are sticking together. So they will have a higher amount of DNA. These cells can be the clumps of 4 cells, 5 cells, 6 cells. So these are clumps, and sometimes it is possible that these are aneuploid cells which are having higher amount of DNA than 92, or there is some problem in the cell cycle, right? So let's see what kind of situation, original situation, we can get an experiment in an experiment. So the answer is simple. What we have done, we have taken T cells which are not dividing. So once they are not dividing, they are having G1 peak. They are not doing replication, they are not undergoing cell cycle, so they are giving us only one peak, that is 46 DNA molecule. So we are calling them as unstimulated cells. Now once we stimulate them with any stimulator, they will start doing cell division. So you can see you are getting all three: the peak of G1, the peak of G2, and the area of S. So software calculates area under the curve to give us the information about cell number or percentage of cell. So you can get 60-70% in G1, 10-15% in S, 25-30% in G2. So you can have distribution of cells in different phases of cell cycle during cell culture or during your experiment. Now, the biggest challenge for this technique is, if I look at this graph again. So if I think about this graph, the challenge I am having here is, I don't know the boundaries of three phases. So I don't know where the G1 is ending and where the S is starting, because the areas are overlapping with each other. Similarly, where the S is ending and where the G2M is starting, I don't know, because areas are overlapped. So to calculate the percentage of cells in G0, G1 phase, S phase, G2M phase, people use different types of mathematical curve fitting or mathematical modeling. Will not go into the details of those mathematical models, but I will be explaining them in brief. The first is the peak reflect method. So we have a statistical quantity which is called as mode. What do you mean by mode? Mode means the parameter which is having highest frequency. So let's say in a class we are having 50 students. Out of 50, 40 are wearing glasses. So the mode of wearing the glasses is what? 40, right? So the system, the model is calculating the mode of G1 phase, okay, and calculating the area on the left side of that mode line. So the central line is for the mode and we are having the area left. Then the system is making a mirror image of this left area towards right also. So we are having this left, the similar amount will be selected on the right, and then the area under the curve will be calculated. For G2, the mode is calculated, the central line, the highest number of cells. Then the area towards right is calculated, and then it is mirror image or reflected towards left. So that completes the G2 peak, and then the area under the curve is calculated to understand the cells per peak or cell per phase. Then remaining is automatically calculated as what? S phase. That is called as peak reflect. The second is rectangle method. In this, in the S phase, rectangles are designed, rectangles are created, and these rectangles are extrapolated till the peak of G1 and the peak of what? G2. So area of all these rectangles is equal to percentage of cells in S phase. Remaining on this side is equal to percentage of cells in G1 phase, and the remaining area on right side is equal to percentage of cells in what? G2 and M phase. So this method is called as rectangle method. The first was peak reflect. The third is S fit. Here a polynomial equation is utilized to create an area which is calculating the S phase, right? And then remaining area on left is equal to G1 phase, and remaining area on the right is what? Remaining area on the right is what? G2 phase. Then we are having SOBR method. In this method, we are having mathematical tools to create Gaussian distribution around the S phase. And once the Gaussian distributions are created, then area of these distribution is calculated. That is equal to percentage of cells in S phase. Remaining area on the left is G1. Remaining area on the right is what? G2. So there are multiple approaches and all of them are giving good results. But we are lucky that we don't have to do it on ourselves right now, because the softwares like Flojo can calculate it. So the Flojo is having the Watson pragmatic method, which is using the polynomial distribution thing or binomial equations, and it is having JET method, which is using SOBR and automatically calculating the percentage of cells in G1, S and G2, right? But we are not that much concerned about mathematics. We just have to understand that we can calculate the area under the curve. The software will do it and will tell us how many cells are in G1, area under the curve, how many cells are in S, and how many cells are in G2. Different mathematical models to do it, but software is using nowadays Gaussian distributions and polynomial equations. So these pictures are credited to a very nice book on flow cytometry called as Flow Cytometry First Principle by Alice Lombardi. And if you go through this book, you will find different applications of flow cytometry explained in easier manner using less amount of maths, right? So they are logical, they are easy to understand. Now we take this knowledge of flow cytometric analysis of cell cycle into the experimental situation where cells are treated with different type of drugs. Okay, so here at 0 hours, cells are given a drug which is called as platinum carboxylate. Okay, and if we look at the result, at 0 hours, at 5 hours, at 16 hours, and at 24 hours, you can see that the amount or the percentage of cells in G1 and S is decreasing, and on G2 it is increasing. So you can say the cells are ultimately accumulating into what? G2. It means they can pass through G1, they can pass through S, but the cell cycle is arrested or blocked by this drug in what? G2 phase. So we can say that platinum carboxylate is a G2 blocker, okay, or blocking the cell cycle at G2M phase. Very simple to understand. At 0 hours, all three phases are there. At 5 hours, you can see some cells are dying also. They are undergoing apoptosis, right? Then you can see that the intensity of the peak or the sharpness of the peak for G1 is decreasing, or S is decreasing. Cells are accumulating in G2, and finally you are having a sharp area or peak defining G2. So you can say that this particular drug is what? A G2 blocker. Now, here in these two pictures, cells are treated with a folate inhibitor. So folate is a very important component of replication. So if we inhibit that, then what will happen? Replication will not occur. Cells will be arrested in what? S phase. They will not be able to cross the S phase. So these are untreated cells. We are having all G1, S, and G2. And here the treated cells after 24 hours, you can see that the cells are mainly in G1, G2 is completely lost, and cells are doing abnormal DNA replication or incompleted DNA replication in what? S phase. So it is the drug, the folate inhibitor, is blocking the cell cycle in what? S phase. Here in these two pictures, cells are treated with imatinib, which is a G1 blocker, which stops the cell cycle at G1 phase. So at 0 hours without treatment, we are having all three peaks, G1, S, G2. But you can see that after 28 hours, we are not having any S, we are not having any G2. Cells are getting accumulated in G1 phase, and ultimately they will die by apoptosis or by different responses towards cell cycle arrest. So we have learned that we can calculate area under the curve, we can calculate the percentage of cells in G1 phase, in S phase, and in G2 phase. We have understood that how the graphs will look like if the cell cycle inhibitors are blocking the phase G1 or the S or the G2. So we have understood both the concept, graph, some part of mathematical modeling, and applications also. So before ending this video, I will be explaining the brief protocol, how we perform this assay, how do we do the pipetting, and what steps are important. So we prepare the single cells and we fix the cells. Fixing the cells is done by using 70% ethanol, some cases in aldehyde and all. So people will ask this question that why we have to fix the cells. The answer is very simple. Generally these DNA binding dye like propidium iodide, they are pumped out by live cells. So if we want our dye should go inside the cell and should stain the DNA, then cells should be fixed. Otherwise we will not get any fluorescent signal. The dye will come out in the media, in the liquid again. It will be thrown out by the cells, by the live cells. So it is important to fix the cells. The fixation of cells should be done at drop by drop method. Means the single cell suspension you centrifuge, you create a pellet, and then you mix the 70% ethanol, you add a drop, you vertex it, you add a drop, you vertex it, you add a drop, you vertex it, you add a drop, you vertex it. So it is a carefully done process where the ethanol or the fixing agent should be added drop by drop because of two reasons. One, fixation will be better. Second, clumps will not form. If we are adding the alcohols, suddenly it is a dehydrating fixator. So there will be cellular clumps and we will not get the proper profile of what? Cell cycle. Third, after the fixation, we have to treat the cells, the fixed cells, with RNAs. The answer is simple, because these DNA binding dyes like propidium iodide can bind to secondary structure of RNA also. So that can create disturbance in our cell cycle profile. So it is important to get rid of RNA molecules, and to do that we are using what? RNase treatment. Then we are finally staining with DNA binding dye propidium iodide. Again, here, just before putting the tube in flow cytometer, you have to add the dye. We cannot incubate with propidium iodide for long term because with time the fluorescence increases and that can give us false signals also. So you just have to add the drop and then you have to put the cells in the flow cytometry, and after that you get the graph, and after that you do the complete analysis of cell cycle. So guys, I hope you have understood the basic principle, how we are doing that, what is the fundamental behind it, what is the brief protocol, what kind of different situation can arise if we are blocking the cell cycle at different stages, and a basic idea about different type of mathematical models or curve fittings to calculate the percentage of cells in G1, S and G2